Welcome back, all participants. Before we proceed to the next session, allow me to briefly inform everyone again about Q&A. If you have any questions, please leave them in Q&A box located on your right side of the WebEx screen, and will share your question to speakers after each presentation ends. And if your question is specific to any speakers, please also mention. Now, the next session, which is our session number 2, will be dedicated to the topic of bioinformatics and data science, unlocking challenges and opportunities. And we are pleased to have Dr. Yuji KOHARA from the National Institute of Genetics of Japan to share this session. Dr. KOHARA research interests include developmental biology and genome biology and molecular biology, and he has worked with National Institute of Genetics since 1989 and currently a project professor of Advanced Genomics Center at NIG and also a director of Database Center for Life Science Research Organization of Information and Systems, Japan. So Professor Yuji KOHARA, the stage is yours.
Yes, thank you very much. Good afternoon, everyone. Now we would like to start the session 2 on bioinformatics and data sciences. So as emphasized by the speakers in the morning session, data is very important, and data handling management is also very important. The data associated with bioresources is very complex, and the kind of the information is very wide. So the connection with this information is very important to use very effectively, more effectively. So the amount of data on the bioresources is enormous, increasing. So today we have 2 experts in the field. May I ask the first speaker, is Shoko Kamoto here? I can't see her name on the participants.
She is here, but
She's here. Okay, thank you.
We cannot have her on the screen, but
okay, so
we can open her mic for her.
So the first speaker is Dr. Shoko Kamoto from National Institute of Genetics, Japan. She received her PhD from Osaka University for gene expression studies. But after that, she moved to the field of databases and joined the database center in the research organization for information systems. And then she moved to National Institute of Genetics, and there she is responsible for the whole database of the National Bioresource project in Japan. So she's now working on various aspects of database management. Okay, so Dr. Kawamoto, would you please your presentation, okay? Yes.
Excuse me?
Yes.
Can you speak? I'd like to present using my own slides.
Sure.
But I can't share. I can't share from my computer because I don't presenter mode, only just a participant. So
okay,
you are in the not in the panelist.
Yes, not now. I'm not panelist, please change my status to.
Sorry,
So
may I say something? Because there is a technical problem, Dr. Shoko cannot be the panelist right now. So maybe would it be possible to have the next speaker first? And Dr. Shoko, could you leave the meeting and come
okay.
back in again?
I'm sorry.
Sorry for that.
So Dr. Ma, is it okay to start with?
Dr. Ma.
Yes, yes, it's good.
Okay, thank you. So then the second speaker can pass, and I'll introduce very briefly the Dr. Ma. Dr. Juncai Ma received his PhD from Department of Bioresources of Mie University in Japan. And now he is director of the Microbial Resource and Big Data Center in the Institute of Microorganisms, Chinese Academy of Science. He is also the director of China National Microbiology Data Center and World Data Center for Microorganisms, and a board member of World Federation of Culture Collection, WFCC. And the title of his talk is Towards the Asian Comprehensive Catalogue of Biological Resources in the Post-Pandemic Era. So could you start, Dr. Ma?
Okay, thank you, Chair. So for the organizer, would you like to transfer the permission, allow me to share my PPT file? Okay.
Okay.
Okay, just a moment. Can you see my PPT file?
Yes.
Okay. So thank you, Chairman, for your nice introduction. I would like to give the introduction for the current development of the ANRRC catalog, and also I would like to introduce our the current activities of our data center. So now for the ANRRC catalog, so we have this kind of the online phase. So for everybody, you can search the ANRRC catalog. You can input any of the strand number. You can input any of the scientific name to search ANRRC catalog. At this moment, the major information is in the microbial resources field. So for the member collection, the ANRRC catalog, now we have the 2 collections from China, CGMCC, FCCHB, and the 2 collection Nbrp JCM from Japan, and the 5 collections from Korea, and also we have collections from the Philippines, from Thailand, and from Vietnam. So totally we have 12 collections from 6 countries and regions. So totally about 8,000 strain in the ANRRC catalog. So we have the strain statistic for each member collection, how many strains they provide in the ANRRC catalog, and how many number of the strains for the bacteria, for the fungi, for the bacteria, about the 40,000 strain information in the ANRRC catalog. So for the user, you can search ANRRC catalog, then you can get strain number, strain information, and then you can buy the strains from our ANRRC member collections. For example, for the GCM, you can click on this particular strain number. You can get the isolation information, and for our data center, we provide the global information related with this particular strain number, such as global publication, patent, sequence information, and so on. And for our data center now, we are the World Data Center for Microorganisms. We are the information center for Bioresources project in the Chinese Academy of Sciences. We are also the China National Microbiology Data Center. So for the WDCM, we are arranging the global catalog of microorganisms, global cooperation. So now we have 145 collections from 51 countries join this global cooperation, about 524,000 strands in the global catalog. So you can search the global catalog, you can get the strand information worldwide. For any of the strand in global catalog, we have this kind of information for this particular strand number globally, how many paper, how many patent, how many sequences, and globally which scientists from which country they are studying this particular strand number. And for the collection, once they join the global catalog, we can give this kind of the statistical report. For example, the DSMZ, one of the best European microbial culture collection. Once they join global catalog, once they provide about 30,000 catalog information strand number, so we can do the data mining for DSMZ. In the past 30 years, every year, globally, how many scientific papers published using DSMZ strands? So we can provide this kind of the report for many of the microbial collections. And the data standard is also one of the important issue. In the morning session, Professor Li introduced ISO TC 276. And in this ISO system, we developed the microbial data standard. So last year, ISO officially published this data standard, microbial data standard. And WDCM, now we are also running the 10K type strand sequencing project. We welcome any of the microbial culture collections worldwide. You provide the DNA or strands to WDCM. We do the sequencing. WDCM cover the sequencing cost. So now we have 25 microbial culture collection from the 16 countries. They provide the DNA or strands to us. We have the ATCC, we have the BCCM, and the CBS, and the CIP Institute Buster. And from Asian country, we would like to say thank you very much. From Japan, we have JCM Nbrp. From Thailand, also we have the TBRC, TISTER, and Korea, we have KCTC, and so on. So thank you very much for your great contributions. And now the website for this sequencing project now is open to the public. Also data is open to the public, and also data analysis services is open to the public. So you, through this website, you can have the type strand information, species information, taxonomy information, and also sequencing result for the annotation information for the type strands. And we have not only a genotype information, but also phenotype information. So we opened to the public. And in Chinese academic sciences, we are the information center for the Bioresources project. So in this project, we have 42 CAS institutions, about 76 different kind of the bioresources collections. So totally about 25 million different kind of the biological resources in CAS. So every year we have our every new catalog, every new version for the catalog of bioresources in Chinese sciences. So last year we opened about 7 million data online. And about 2 years ago, Chinese government set up a so-called National Scientific Data Center system. So totally now we have 20 scientific data center in China. We microbiology data center is one of the 20. So those kind of the national data center, we are in charge of the for any of the national project. Before the project finish, the PI must provide the data. You have to open the data. You have to provide the data to the national data center. So in the COVID-19, we developed the COVID-19 National Science and Technology Resources Service System. So we opened the first COVID-19 microscope, virus strain information, and the primary information to the world. And also we helped the Chinese scientists open the data to write the scientific papers for the COVID COVID-19. And quite a lot international media reported our National Microbiology Data Center, NMDC, published the microscope pictures, strain information to the world. And also we developed the RDF-based, RDF knowledge-based database systems for the COVID-19. So we put strain information, global paper information, sequence information, structure information, protein information, antibody information into one database system. So we have this kind of the virtualization to the public and also the data analyze. And also we put the past about 7 major diseases like SARS, like COVID-19. So we put all information together so the user can show the scientific data, can do the analysis through our system. And recently, we also developed the evaluation and the pre-warning system for the COVID-19. So we put global, we have the cooperation with GSide and NCBI. So we put all the strain information, sequence information together. We do the data analysis. We provide the data searching function to the public, and also we do the analyze variation information for the genome precision, how the genome precision change, and what kind of the efforts will be, whether the efforts big enough for the primers or not. So we provide this kind of the scientific data support for the government for the control of the COVID-19. And also we provide this kind of identification and functions. Online user, you can input any of the sequences, information, that you can get the identification result. And also we, for each kind of the variation, like delta, beta, alpha, so we have this kind of the data source open to the public. So this data platform was just opened to the public, and 2 weeks ago, this paper, scientific paper, is online in the nucleic acid research. And also in the morning session, some colleagues talked about the young scientist development. And we are very happy for the Dr. Lin-Wen Wu. So she just received the WS data awards, which is a very important award in the scientific data field. So finally, I would like to see for the future Asian catalog, we hope we will have more and more collections from the more and more country. Then we can develop not only the microbial catalog, but also more kinds of the biological resources open to the public. And also we hope the Asian catalog can have not only the isolation information, but also more functional data like sequences and so on. Maybe RDF related functions will be available in the public. And also more and more data analyze functions should be available in the Asian catalog, and also virtualization functions for the big data is also important. And finally, our institute just developed a new journal called MLife. So we welcome, so basically all kind of all fields related with microbiology is covered in this new journal. So we welcome our Asian colleagues submit your next paper to the MLife. So finally, I would like to thank the great contributions from the cultural collections worldwide, and also strong support from the Chinese academic sciences, especially International Bureau, the Bureau of the International Affairs, and also Ministry of Science and Technology. And also I'd like to thank the great works from the staff of my team. Thank you very much.
Thank you very much, Dr. Ma, for the very brief introduction for such a great work. So is there any question, comments? In the Q&A, just one. Is that correct? I can't see. How was the situation about ABS in the culture collection? Can you see?
Okay.
The,
For the ABS, I think it's a very big issue. So for our sequencing project, we also receive the strands from the cultural collections worldwide. So we ask any of the collection, before you send your DNA or strands to us, you need to follow your national laws for the ABS. I think Filippo this math is experts in this field.
okay, thank you very much. Any other question, comments? So as to your virus databases, so you said that you are make it open. So have you published in the make open in the INSTDA?
Yes, for basically for all of our database is open to the public. All data is open to the public, and also the sequencing result for the TAP strand sequencing result. The data is also open to the DDBJ gene bank, and then also.
It's very nice.
Yeah.
Thank you very much. Or any other question or comments? Okay, if not, it's just in time. So thank you very much. Then I would like to move to the next person speaker. Are you ready, Shoko? Okay. So,
Oh, can you hear me?
Dr. Kawamoto, please.
Can you hear me?
Yes, please.
Okay, I'm sorry for the trouble. I can't to be a presenter, so organizer operate my presentation, please. Hello, everyone. My name is Shoko Kawamoto from the National Institute of Genetics. I'd like to thank the President, Dr. Lee, and organizer and the participant and the chairperson for giving me the opportunity to speak this session. First, next slide, please. First of all, I'd like to introduce my institute. This is a picture of my main entrance. The National Institute of Genetics was established in 1949. Next slide, please. In our institute, there are 3 centers related to databases and information. The most famous center is the DDBJ, member of INSTDA with NCBI and EBI. DDBJ also operates a supercomputer used by many Japanese researchers. In addition to that, we have the Advanced Genome Center, which conducts large-scale genome sequencing. In other words, our institute has a strong advantage in genomics and informatics. Next slide, please. Taking this advantage, our institute has been conducting analysis of SARS-CoV-2 since last year in cooperation with the Shizuoka Prefecture. Patient RNA samples sent to our institute are sequenced by NGS, analyzed by supercomputers, and the sequence will be shared publicly to the world through DDBJ. Next slide, please. Next slide. In this way, our institute is characterized by its facility for genomics and bioinformatics research. We are also working on the dissemination of biological resources. The Genetic Resource Center plays the activities for Bioresource. This center was established in 1997. There are 2 divisions. The Bioresource division takes responsibility for development, preservation, and distribution of bioresources of various organisms. The database division is developing databases for genetic resources, especially on NBRP information, which shows the next slide. Next slide, please. NBRP, National Bioresource Project, started in 2002, funded by Ministry of Education and Science Japan, and this project continued 20 years long. There are 32 resource projects and 36 kinds of organisms available. From last year, facility of human pathogenic viruses have been established to prepare for pandemic like COVID-19. Next slide, please. This slide shows the location of organizations participating in NBRP. There are over 60 organizations related to the project. Next slide, please. This slide shows the current NBRP portal's website. By accessing this site, you can get all the information about the NBRP project. The URL is nbrp.jp. Next slide, please. On the resource search page in this portal site, you will find links to the resources participating in the NBRP by each species. When click on the link of the resource name, and user can access the databases of the organisms you want to see. Next slide, please. And this slide shows the part of the various database pages providing our information center. Through these websites, we manage and publish about 100 databases. Strain information database. In addition to the strain information databases, there are many services and information such as net shopping sites, genome browsers, and so on. Next slide, please. This table shows the number of available resources in each resource project. Each number contains wild-type strains, mutants, cDNAs, EST clones. When counted at the species level, it contains about 8,700 species. General resources such as mice, culture cells, and plasmids are managed by Riken Bioresource Research Center, and others are distributed public institutes such as universities and research institutes. Among these, Medaka, Sayona, Japanese macaque, morning glory, and cellular slime molds, silkworms are unique Japanese resources. Next slide, please. This slide shows the service that enable you to search 6.5 million resources managed by the NBRP project. The search result is directly linked to each resource site. However, there are several problems with this system. Example for it only returns hits with an exact text match. This because different terminologies are used in different domains of research, making it difficult to search for terminology about different organisms. Next slide, please. When we break down the difficulties related to search and databases, it can be seen these problems. Problem 1 is evolutionary orthologous genes have different names. And problem 2, the same phenotype is described by different expression in different languages. Problem 3 is the ecological data is lacking or insufficient or described by different expression. Next slide, please. In order to solve these problems, bioinformatics and semantic web approaches are needed. And today, I'd like to talk about the semantic web approach that we are trying now. The semantic web approach requires three things. First, a controlled vocabulary and ontology. Second, a data format specifically for the semantic web. And third, a query language to manipulate the database. In the next slide, I will explain about the data formats. Next slide, please. The data format for realizing the semantic web is named RDF. RDF represents information in a series of three-part statements that comprise the subject predicate. This slide shows the example of the statement Bangkok is the capital of Thailand. A model for encoding semantic relationships between items of data so that these relationships can be interpreted computationally. And also RDF databases are often referred to as graph databases. Next slide, please. This slide shows the previous sentence extended with other information. The subject and the object can be thought of as two nodes of the graph with the predicate defining an edge between them. Predicate and object classes should be described in ontology to connect with other databases. A traditional relational database might split attributes into separate tables. In RDF databases, all these data points belong to the same interconnected graph, which allows users maximum flexibility in deciding how they wish to query it. Next slide, please. In order to convert the existing bioresource database into RDF, we developed an ontology to represent the data in RDF named BRSO. BRSO has properties for taxonomy, genotype, and phenotype, which are the basic attributes of resources. And a feature of BRSO is that it is structured to match the data of the resource center. For example, we can get the information of the depositor of the resources and the distributor of the resources from the RDF graph database. Next slide, please. Using this BRSO, we are currently working on RDFing data from algae, silkworm, yeast, nematode, and zebrafish. And this slide shows a website developed by using RDF data of Nbrp algae and its RDF data schema and query language. I'm sorry, this is all I have to show you today in the way of results, but as the data increases, it will be easier to handle the data and develop the website because all resource data will be described in the same format. And finally, I think it will be possible to handle databases of different organisms in an integrated manner. And I hope to show the results at the next ANRRC. Next slide, please. And finally, I will spend a little time to introduce you to our other database. This slide shows the capture of the Bioresource research paper database named RRC. Review of papers published by using NBRP resources. Currently, about 46,000 articles are registered in this database. You can search by resource name, journal name, and authors. Next slide, please. This database allows you to retrieve data by the nationality of the institution to which the first author of the article belongs. And here are the results for Thailand. The number of papers which first author's affiliations are in Thailand using Bioresource in Japan is total 220. When I extracted papers on RJ from the papers from Thailand, I found several papers on bioreactors. As you can see, the paper information can be used to help building research plan and find resources related to the research topic. Next slide, please. Fine. This is the last slide. Finally, the development of BRSO is a collaboration between Dr. Masya of Riken BRC and the BCLS. For the development of the research paper database, data were provided from the Riken BRC and the Nbrp Resource Center. Thank you very much.
Thank you very much, Dr. Shoko Kamoto. The time is limited, but there's one question from the Q&A. Can you see the Q&A? How do you maintain the databases? Do you have any cloud system for the database?
Yes, we maintain the database in-house server, and but we plan to use cloud server.
Okay.
Gradually.
So any other question or comments in the board? Okay, if nothing, okay. So if not, I'd like to close this session. Okay. Thank you very much for distinguished two speakers for your great talk. Thank you very much.
Thank you very much, Dr. KOHARA, for hosting this session. So next, we would like to move to the next session on the how plant research resources support BCG economy. Please allow me to introduce the chair of this session, Dr. Masatomo Kobayashi. Dr. Kobayashi was a research scientist at Riken since 1985, and he has got a doctorate degree from Tokyo University and did the postdoc at University of California, US. Dr. Kobayashi is currently head of Environmental Plant Division, Riken Bioresource Center. Dr. Kobayashi, the stage is yours.
Good afternoon, everyone. Thank you for your introduction. Now we start session 3. How plant research resources support BCG economy. So, as you know, National Bioresource Project in short, NBRP, is running in Japan. Today, we have two speakers from NBRP. The first speaker is Dr. Hiroshi Abe from Riken BRC. Let me introduce you his career. Dr. Abe obtained doctoral degree from Tokyo University of Agriculture and Technology in 1998. And then he spent 4 years as a postdoc in Japan International Research Center for Agricultural Sciences. Then he moved to Riken BRC. He joined the Experimental Plant Division as a research scientist and engaged in the Arabidopsis project of NBRP since 2002. Today, he will talk about the features of Arabidopsis status of Arabidopsis project and his experience in utilizing Arabidopsis in BCG. Dr. Abe, please.
Hi, so can you hear me?
Yes.
Okay. Okay, so I start sharing my slide. So share. So can you see my slides? Okay.
That's fine.
Okay. So first I'd like to say thank you for organizers giving me a chance to talk about our recent activity in such a great and wonderful conference, so ANRRC 2021. So my name is Hiroshi Abe, working on the RIKEN Bioresource Research Center. So I belong to the experimental plant division, and so we are the one of the three biggest Arabidopsis resource center in the world. So especially we are the core faculty in Asia. So this picture shows the Arabidopsis plant. So as you know about it, I hope Arabidopsis plant is the most studied and the most advanced experimental model plant. And so this plant is essential for the plant science. But so why Arabidopsis plant? So Arabidopsis plant has many good convenient features for the research. So small size, less than 30 centimeter tall, and so growing fast. 2 months is enough to get the next generation seed. And so growing easily under the fluorescent lamp and also LED lamp. So we can transform the Arabidopsis plant using the very simple and very easy protocol. And so small genome size is also a very good point of the Arabidopsis plant. So this plant is the first plant to get its complete genome sequence in 2000, so 20 years ago, about 20 years ago. And after that, so many kinds of the comprehensive genome resources have developed one after another, such as the comprehensive gene knockout mutant. So natural accessions is also a very powerful tool to understand how plant adapted to the many stress conditions. Of course, so many kinds of the genetic mutants is also identified and so available from the stock center. Of course, from the RIKEN BRC. So we have collected and preserved more than 800,000 Arabidopsis resources from the Japanese scientific community. So it contained, of course, Arabidopsis seeds such as the natural accessions and also genetic mutant and gene knockout mutant, and also transgenic lines. And we also collected the Arabidopsis seed pool for the mutant screening. And so we also collect and preserve the Arabidopsis cell cultured cell lines and also full-length cDNA clones. So we distribute so many kinds of the Arabidopsis resources to the researcher around the world. So from 2002, we have distributed around 60,000 resources to the international scientific community. So we have distributed our resources to 50 countries, and 57% of the distributed resources are in Japan, and so the other 43% is for the overseas. And so nearly 90% of the distributed resources are for academic users. So in the bioresource operations for us, so quality control is one of the most important missions. So presence of the defective bioresources is a very big problem to promote the research and so to promote the science. So we aim to decrease and reduce the defective bioresource to the 0% using the strict QC, so quality control, at the time of the deposit and at the time of the distribution. So this window shows the example of the contents of our quality control. So specific PCR tests for the transgene in the transgenic resources. And of course, so sequence analysis and also genotyping using the SNPs and so SSLP markers. And so we also use the marker genes such as the major antibiotic resistance gene and also major marker genes, reporter gene for detecting for the mistake and the contaminations. Okay, so when we find the defective bioresources at the time of the deposit, so we contact to the depositors and so discuss the response and so implement remedial measures. And so we also collect the bioresource information and so notify these updated information to the user using the QC report, quality control report. And so we found the defect with the 40% of the Rhabdopsis transgenic seed. And also we found the defect with the 27% of the Rhabdopsis full-length cDNA clones. Okay, so this is a case of the Rhabdopsis full-length cDNAs. So when we get the order, so we start sequencing, and so we compare the information of the sequence of the full-length cDNA to the information of the Rhabdopsis standard annotation. So most of the defects are the 5' deletion and so 3' deletion, non-synonymous mutation, and so internal deletion insertion. So anyway, so when we find the defect, so we start find for the alternative clone with no defect. So in 70%, so we can find the alternative clone with no defect. Then, so we report the QC, so quality control result to the user and suggest the alternative clone, if any. And so we wait for the answer from the user, and so we distribute the full-length cDNA clones. But anyway, so we think quality control is very, very important for our activity in the bioresource operations. Okay, so let me change my subject from bench to bountiful harvest. So from bench to bountiful harvest. So this is a recent slogan, so decided by the multinational Rhabdopsis steering committee. And so we are also on this committee. So Rhabdopsis plant is just a weed, so not a vegetable crop. So we cannot eat the Rhabdopsis plant, but Rhabdopsis research can contribute food and environmental issues and so contribute to the SDGs. So today I'd like to talk about our trial to develop the insect pest repellent starting from the Rhabdopsis research. So recently, the agricultural damage caused by the insect pest is becoming more serious. And one major reason is global warming. So pest management is mainly controlled by the insecticides, but recently insect pests with the insecticide resistance are emerging. And so this is a very big problem in the world. So one such typical insect pest is the thrips. So thrips is called to the elite of the insecticide resistance. So one of the most serious insect harbour difficult to control, this is also thrips. And so thrips is the insect pest and also virus vectors. So thrips can transmit the plant virus from the plant to the plant to plant. So economical damage by the sweeps is very serious in the world. So hours of Arabidopsis research can simply suggest that activation of the plant defense system regulated by the plant hormone jasmonate JA is very important for the plant resistance to the sweeps. So JA regulated plant defense is very important. So one of the very good advantage of the Arabidopsis plant is the existence of the so many kinds of the mutants. And so in this case, so we can use the no defense mutant named KOI1 mutant. And sweeps can attack the wild type plant, normal plant. And the wild type plant can still grow up and producing the flower and also producing the next generation seed. But so in the same condition, so this no defense mutant fed everything by the sweeps. So there is very big difference. And so interestingly, sweeps increase their population on the no defense mutant as compared to the wild type plant, as shown here. So we question to the sweeps, so which do you like? So which do you like, normal plant, so wild type plant, or so no defense Arabidopsis mutant? And so this is our answer of the sweeps. So most sweeps were attracted to the Arabidopsis mutant. So we thought that so we can control the sweeps behavior using the plant defense system. So we checked the treatment of the JA, and so we asked to the sweeps again. So which do you like? So JA, so treated the plant, Arabidopsis plant, or so water treated Arabidopsis plant. And so JA treatment can enhance sweeps avoidance in Arabidopsis plant. So JA treatment can activate the plant defense system, and so activated plant defense can repel the sweeps. So this is a very good point. But so JA treatment inhibit the plant growth. So JA itself is not good for practical application. So we next search and we next analyze the JA analog. And so one JA analog named PDJ, so prohydrojasmone, is very interesting. So PDJ is originally developed in the Riken Institute about 20 years ago, and now in commercial use as the plant growth regulator to improve the fruit color of the apples and the grapes. Okay, so PDJ treatment also activates the plant defense like JA. And so activated plant defense can repel the sweeps. And so importantly, so PDJ treatment has no effect, so no agronomic effect for the plant growth. So PDJ is one good candidate for the practical application to control the sweeps behavior. So then, we changed the plant from the Arabidopsis plant to the vegetable and flower crops. And so PDJ can also repel sweeps in many plant species, like in Arabidopsis plant. So this picture shows the PDJ test in the greenhouse tomato cultivation. And this is the example of one data. So blue color means the insecticide treatment. So insecticide treatment can reduce the sweeps population. And so red color means the PDJ treatment. So PDJ treatment strongly reduce the sweeps populations. Okay. So in this spring, so we get a proposal of the PDJ as a repellent from the Japanese government. And so now we try to develop the system for the sustainable and so environmentally friendly pest control system based on the PDJ. And so our goal is the agriculture in harmony with living things. So this image is our SDGs. Okay. So this is my last slide. So summary. So with the variety of the resources, Arabidopsis is well suited to understand life phenomena and to develop and implement research results. And so quality control to reduce the defect of resources is very important to maximize the benefit of the bioresources, benefit of the Arabidopsis resources. And so JA analog PDJ control sweeps behavior as agricultural concern, and also JA analog PDJ was approved as the sweeps repellent in the tomato in this spring in Japan. So thank you for your attention.
Thank you for your talk, Dr. Abe. I cannot find any question in Q&A. So just I would like to ask you one comment, your comment. You applied Arabidopsis for the development of pest control technology. Is there any research field in which utilization of Arabidopsis is effective in the development of novel technologies for agriculture?
Thank you for your question. So I think there are many possibilities. So for example, so Arabidopsis research have found so many several important compounds to confer the environmental stress resistance, environmental stress tolerance in the plant. And so in that case, so Arabidopsis resource is very helpful. And it's also effective to use the Arabidopsis mutant with the complete same genetic background to understand the complexity of the multiple interaction in the ecosystem. So that kind of the analysis, so Arabidopsis mutant is very powerful, I think. And so this kind of the analysis in future is very important. And maybe it could be help us to find the important gene, the target of the genome editing, maybe. So there's many possibilities of the Arabidopsis, I think.
Okay, thank you for your comments. And now time is up, so we would like to move to second speaker.
Thank you very much.
Okay, the second speaker is Professor Yutaka Sato from National Institute of Genetics. Dr. Sato got his PhD in 1998 and then became associate professor in Nagoya University in 2003. Since 2016, he served as a professor in National Institute of Genetics, which is the core facility of RICE in Nbrp. In his talk, he will introduce his efforts to establish the resource infrastructure of wild rice, which is useful for promoting BCG, I believe. So, Professor Sato, please.
Thank you for your kind introduction. Let me start with sharing slides. Can you see the slide?
Yes, that's fine.
Okay, so I will talk about the development of molecular genetic research tools of wild horizon. Okay, so I'm in charge of operating Wild Horizon Genetic Resource Center in National Institute of Genetics in Japan. And wild ORIZA genetic resources in NIG is collected during 1950s to 80s by Professors Oka and Morishima. And we are still maintaining those materials. NIG holds wild ORIZA resources over 1,700 accessions covering 21 wild ORIZA species from all over the world. As you may know, wild ORIZA species are the origin of cultivated rice. For example, ORIZA ripogon is an ancestor of domesticated ORIZA sativa, which is food for rice, food for us. And also, there are another domesticated species of rice, which called ORIZA glaberrima. ORIZA balsi is a wild accession and ancestor of ORIZA glaberrima. So, wild ORIZA genetic resources are very important to understand the process of domestication, or also wild ORIZA genetic resources are important for the gene pool for future breeding, future use in breeding of cultivated species. This is one example of wild ORIZA species, ORIZA ripogon, which is ancestor of ORIZA sativa, and they live in along with water-rich environment, for example, small rivers or something like that. And they originate from mainly from South Asia. Purpose of our activity is to promote the usage of wild ORIZA genetic resource for research, especially for biocircular green researches, I think. And in order to accomplish this purpose, we are trying to obtain genome information and release its information from our database called ORIZA base. So if you're interested in, just Google ORIZA base, then you will reach to our database. And another way to promote the usage of wild ORIZA genetic resources is to exploit molecular genetic research tools. By combining these two efforts, I think we can accelerate the wild ORIZA researches. Then it also promotes more researchers to attract wild ORIZA genetic research. So this kind of circle will, if this kind of cycle operates, then it will lead to the promotion of the usage of wild ORIZA genetic resources. So, in order to exploit molecular genetic research tools, we first tackle with the development of genetic transformation method for wild ORIZA research. This is a very simplified procedure to the genetic transformation in generally in rice. We introduce rice technology, we modify methods to the genetic transformation of cultivated rice to apply, in order to apply wild ORIZA genetic transformation with small modification. So first, we put mark 10 flowers bloomed like this. I think we should use on there. So like this, we put a mark 10 the flowers bloomed. Then after 8 to 11 days, we harvest ovaries from these flowers. Then excise immature embryo. Then we put agrobacterium onto the immature embryo, then culture on the medium, and then we transfer the regeneration method to get a regenerated plantlet. This is a basic method to the genetic transformation. And this is it's kind of a process to obtain immature embryo. So we have to collect a huge number of flowers to collect immature embryo with many people. Then conducted culture to test if it is applicable for genetic transformation. So this is a picture of spikelet, and this is a picture of 8 to 11 days after pollination ovary. This is cultivated species. We can stably transform cultivated species, but not many wild accessions are applicable for this method. method established for cultivated species. So we change many conditions to adjust for the transformation of wild ORAISA resources. This is a list of conditions we change to do genetic transformation of wild ORAISA. The first two methods are developed basically for cultivated species such as Japonica species or Indica species. But we have another option to transform wild ORAISA, and we test these 4 conditions to transform wild ORAISA. So we use almost 200 accession wild ORAISA species to test these 4 conditions for the applicability of genetic transformation. And finally, we obtained 19. We found that 19 accessions are good for generation in these conditions. And also, using these 90 accessions, we tested if agrobacterium can infect those culls induced by these 90 accessions, and it turned out that 47 are easily transfected by bacterium by judging from GFP fluorescence. And by using this kind of method, this is one of the examples of the successful case for genetic transformation of ORAISA species. This is the picture immediately after agrobacterium infection. I think this picture took after 40 or 60 hours after agrobacterium infection. Then you can see the GFP fluorescence like this. Then after we transfer these immature embryo into selection medium with antibiotics, then these transformed cells proliferate like this. Then after we transfer these culls onto the regeneration medium to get to shoot. So you can see various small, many small leaves emerging from the regeneration medium. Then after we obtain small plantlet like this, then we can transfer these plantlet onto soil and can grow until maturity like this. And this is a picture of transgenic plants obtained from this method. This is one example of ORAISA brachyansa. This is a GFP fluorescence in leaf. This is a GFP fluorescence of T1 seeds. T1 seeds means the seed set onto the first regenerated plantlet. So by seeing this picture, so transgene is stably transmitted to the next generation. Okay, so we screened ORAISA species which is feasible for genetic transformation by several conditions. And roughly 1/4 of ORAISA accession we have can be transformed by our method. So by using those genetic transformation methods, we can conduct similar experiments such as explained in here. Usually, conventional breeding takes long time because cultivars are crossed with disease-resistant strains or drought-resistant strains or salt or high-temperature-resistant strains or other beneficial strains with other beneficial characters. And many times of backcrossing take long time, then make new cultivars. But this method is maybe takes too much time. So maybe we prefer to have much faster way to get new varieties. So by using wild ORAISA species which have high diversity, including and they live in various habitats, so they may have more disease resistance, or they may have more drought resistance, or they may have more salt or high temperature resistance. By using those wild accessions, we can conduct genome editing to get the character of cultivated species. So it means wild accession can be directly domesticated by genome editing. In order to do genome editing, transformation technology is necessary. So in that sense, our the genetic transformation methods of wild ORISA developed by our effort will contribute to those kind of researches in future. So this is one example of the famous trait of wild ORISA. So wild ORISA shows shattering phenotype. It means many grains drop off immediately after its maturation. So it's not suitable for agriculture. But by modifying 1 or 2 genes, then the famous character of wild accession can be transformed into domesticated type, which does not shatter. So this kind of experiment can be done using our genetic transformation method. So we are very happy to, if researchers use this kind of technology to improve the character of many varieties, including wild ORISA species. And these are collaborators. Thank you very much.
Thank you, Professor Sato. Oh, thank you very much. Please use Q&A function if you have any question. I can't find any question right now, so I would like to ask you one question. Probably it is difficult for rice species to exchange resources between the foreign countries. So what is the most difficult point that you want to provide rice seeds to foreign users? What will be the biggest barrier?
Okay. I think you're asking about the plant clenching procedure, right?
Maybe, yes.
Yeah. Basically, we can send wild ORISA species to many countries, but in some cases, it depends on countries to countries. And I faced one case in Malaysia. When we export seeds to Malaysia, granting office request very difficult to check, difficult procedure to conduct to check the infection of bacterium to the ORISA seeds. We have to prove that there is no infection or carryover bacteria in the rice seeds, but sometimes it is difficult. But in many cases, the clenching process is not so difficult. So basically, if there is a request, we can send our material to everywhere.
I see. Anyway, you try to solve such a problem in every case.
So I faced one difficult case so far.
I see. Okay, is there any question from the audience? If not, then the time is up. So thank you very much for your presentation, Dr. Sato.
Thank you very much.
And, I would like to close the session. For audience, thank you for your attention.
Thank you very much, Dr. Kobayashi, for hosting an excellent session. And now I would like to move to the next session on human research resources in the post-COVID-19 era, which will be shared by Ms. Jane Carpenter. Jane has established the Australian Breast Cancer Tissue Bank and managed the development of the NSTDA Health Statewide Biobank. And currently, she is with the NSTDA Health Pathology and also serves as a biobanking advisor to the Westmead Biobank in Western Sydney. She has been involved with the International Society for Biological and Environmental Repositories, or ISBRS, at the highest level and was a member of the board for 6 years. And Jane also serves on the executive committee and has been a member of ANRRC for over 10 years. Now the stage is yours, Jane.
Thank you. I'm going to have to shout because I don't think my microphone's working too well. But welcome to the Human Biobanking session. We've got two great speakers lined up today, and I know we're running a little bit late, so I will move forward quickly. Our first speaker is Professor Jennifer Byrne, who is Director of Biobanking with New South Wales Health Pathology and Professor of Molecular Oncology in the Faculty of Medicine and Health at the University of Sydney. She has extensive experience in biobanking from both the perspective as a researcher who has employed biospecimens throughout her research career and through establishing, overseeing and networking cancer biobanks. Professor Byrne was the founding chair of the Australian and New Zealand Children's Oncology Group Network of Tumour Banks, and she chairs the New South Wales Health Statewide Biobank Scientific Advisory Group. She's also a member of the steering committee of the Brain Cancer Biobank in Australia and the Victoria Cancer Biobank Scientific Advisory Board. She has led several Cancer Institute New South Wales funded projects across all cancer biobanks in New South Wales, including a collaboration to adapt and implement the first biobank certification program to be introduced to Australia. She's published over 100 peer-reviewed publications, which have been cited more than 3,200 times. And her current research interests include the analysis of human biobank research outputs and the identification of predictors of biobank success, which I think is dear to all our hearts, and the detection and analysis of incorrectly reported nucleotide sequence reagents in the biomedical research literature. So with that impressive array of activities, we're very pleased that she has found time today to present to us, and the title of her talk today is Human Health Biobanking During the COVID-19 Pandemic, the Challenges and Opportunities. So welcome, Jenny.
Jane, thank you very much for that very kind introduction. I'm going to try and I can share my slides, I think. So I think you can probably see that. Them on. There we are. Hopefully you can see my slides. So look, thank you again. I've really enjoyed the presentations today, and I've learned a great deal from listening to everybody. So I'm going to talk a little bit about health biobanking, but before that, I'll just briefly introduce the Statewide Biobank, which is where I'm joining you from today. So as Jane indicated, this is a state-of-the-art facility to support health and medical research in the state of New South Wales, which is the most populous state in Australia. It was a purpose-built biobank that operates largely through a prospective collection model, and I'll explain what that means. We also employ cost recovery to enable flexible service delivery and to expand our services in the future. And our biobank is supported by the Office of Health and Medical Research in New South Wales, and we are operated by New South Wales Health Pathology. So I'll be talking about a few different things fairly briefly in my presentation today. So I'll start by just giving some context to human health biobanking and its contributions to research, because I recognise that many of the presentations today have been about other fields of biobanking, which has certainly been very interesting for me. So a lot of my work is inspired by this kind of result, which was published in this case in 2015 by Friedman et al., which estimated that about 50% of the US investments into preclinical human research supports irreproducible research. So human research has a big reproducibility problem on its hands. And the single major cause of that was estimated to be biological reagents and reference materials, which is clearly where biobanks come in. So biobanking, in terms of supporting human research, enables relevant research because it enables researchers to employ specimens from humans that are likely to more accurately reflect complex disease. It also enables the derivation of other models such as cell lines, organoids, and xenografts. And essentially, human biospecimen availability and quality are major determinants of the research that is possible. So biospecimens essentially circulate through biobanks to support discovery-based research, validation research, and then clinical trials. And biospecimens enter biobanks from clinical trials and clinical care. So there is this kind of circulation of biospecimens and knowledge through these research pathways. There are a number of different biobanking models that are used to support human research, and I'll present those very quickly. So the classic biobanking model is essentially collections that are built around either research aims or available biospecimens. The prospective biobanking model, which has become more popular, is where biospecimens are collected for specific research projects, so for predetermined use. And the aim here is to reduce the waste that's associated with collecting specimens that are never used. Population-based biobanks largely include samples from either healthy or at-risk populations. They are particularly expensive to establish and maintain, and so are currently rare in Australia. Archival biobanks are popular in the sense that they can capitalise upon biospecimens that have been collected for clinical care, but these are not always suitable for research from either an ethics perspective or a biospecimen perspective. And finally, data-focused biobanks are also, I think, something that people are increasingly talking about because they are less expensive to operate, but they have a reliance upon good IT resources. So some current challenges in health biobanking, I mean, some of these are not new. Essentially, planning and running a biobank involves 3 stages: the setup phase, the operational phase, and then a phase where the biobank will either further expand or it may consolidate and wind down. So in health biobanking, the setup phase can be quite variable, but essentially a health biobank can be established largely from facilities and equipment that are present in ordinary laboratories. And it is this capability to establish biobanks at quite a low cost, which combined with this ready access to biospecimens through healthcare, particularly in countries such as Australia with a public healthcare system, that leads to potentially large numbers of health biobanks. So in Australia and many other countries, we have a very fragmented health biobanking sector, and this is likely to contribute to poor visibility of individual biobanks and their under-resourcing and underuse. So in terms of health biobanks, many biobanks are difficult to find because we lack good all-encompassing biobank registers. We often impose burdensome access processes on researchers that want to obtain biospecimens. The biospecimens themselves may lack the right biospecimens or data, and as a result, researchers may need to access multiple biobanks, which increases the burdensome nature of this process. And of course, when biobanks are underused, we have a number of downstream outcomes: wasted resources, including goodwill, which is very important in health biobanking. We have a reduction in capacity for biobanks to recover their costs, to learn and improve from research interactions, and of course, reduced research opportunities, which is why biobanks were constructed. So COVID-19 has obviously had many impacts upon society and different forms of research, but I think they've probably had arguably the greatest impact upon health research. And we published a paper earlier this year where we described some of the impacts upon biobanks, and I'll be talking about some of these with you now. So essentially, COVID-19 has impacted both the supply of biobank resources and demand for biobank services. In the clinical sector, this is because there has been sometimes dramatic changes in clinical research allocation in response to COVID-19 infections, changing clinical research priorities, reductions in clinical trial capacity, which both generate biospecimens and consume them. Patients seeking face-to-face care has impacted upon patients consenting for biobanking, and there can be reductions in-kind support in clinical sectors because of the need to redistribute resources to COVID-19 patients and care. So within the research sector, there have also been quite serious consequences of the current pandemic in Australia. For example, changing in research priorities, reductions in research funding, and an increasing uncertainty for researchers and academics in terms of their careers. This reduction in funding has sometimes directly impacted biobanks and also career uncertainty within that sector. Because we have been subjected to quite extensive shutdowns, there have been problems with accessing facilities, both to conduct research and also to actually operate biobanks. And we've also experienced problems in maintaining complex equipment due to quarantine requirements for international travel. So, of course, every pandemic or every crisis tends to provide opportunities. It just takes a little bit of time to look for them. So within the research sector, of course, we're all meeting online today, and there's been a very rapid transition to virtual meetings, and those can be extended to biobanks. So biobanks can use virtual technology to meet with research clients and reduce the impact of the pandemic and the isolation that has imposed. Virtual research conferences are very accessible and less expensive, which can be important for biobanks in Australia. And we believe that there's a lot of opportunity to create joint conferences between researchers and biobanks to improve biobank use. There are, of course, for biobanks that are able to support COVID-19 research, there have been many opportunities to support COVID-19 projects, and our biobank has been fortunate to be availed of those opportunities. There's also an opportunity, I think, to rethink how biobanks actually support evidence and obtain evidence for that through research, as well as internal activities such as stock management, quality control projects, and also marketing and culling samples that are unlikely to be used in the post-COVID era. We're very interested in gaps in biobanking knowledge, and we believe that there's not enough known about what we refer to as the organizational dynamics of biobanking, essentially how biospecimens and data are employed in research in the real world and by whom, and whether different biobanking models actually operate better for different fields and in different resource settings. And of course, we need to examine the impact of COVID-19 on biobanking success and on research demands and expectations. We've published a paper in 2020 where we talk about the need for biobanks to pay more attention to the outputs that they produce as part of this process. So very briefly, as a summary, small health biobanks are much easier to establish than they are to sustain, and this is likely to lead to health biobank underuse and sustainability problems. The COVID-19 pandemic has certainly impacted both the supply of health biobanking resources as well as demand for its services. But virtual conferences and communications and the support of COVID-19 research have represented important opportunities for health biobanks. And finally, we believe that research is needed to describe how health biobanking supports research to make a case for ongoing financial support of biobanks. So I'd just like to thank the staff at the New South Wales Health Statewide Biobank, the Office of Health and Medical Research, and New South Wales Health Pathology, the Centre of Health Record Linkage here in Sydney, and the members of my research team. Thank you very much for the invitation to speak today.
Thank you, Jenny, for a great talk. Very interesting, as always. I don't see any questions coming up in the Q&A, but I'd just like to ask how you think we can maintain the opportunities. So things like joint research biobanking conferences that, as you say, virtually are somewhat easier to access. How do we maintain that momentum once the pandemic's over and things are kind of getting back to how they were before? How do you think?
I think that's a really good question, and I think in some people think that things are actually not going to return to how they were before, because particularly in terms, look, I think a lot of people would love to travel and see their colleagues, but the reality is that you know, conferences are going to be cheaper to run in the virtual world for some time, and I think that will drive behaviour. But of course, any kind of, as we all know, I mean, the organisers of this conference will know very well, organising any conference takes a long time. So if we are going to think about these kinds of research and biobanking conferences, that's something that I think we need to be doing now, because they're likely to take place in the future. But it's an interesting question. I think, yeah, it's Australia perhaps as an isolated country has perhaps benefited from some of these virtual arrangements as well, because we have to travel long distances, and in terms of the, you know, a carbon neutral economy, perhaps researchers are increasingly thinking that jumping in a plane and traveling, you know, long distances for conferences is probably not going to be sustainable in the longer term.
Yep, great. There's a question to come through on the Q&A. Do you have the exact protocol of how they collect the samples? I'm not exactly sure which samples, because the human biobanks hold a vast array. And how many years are they held for? What type of tissues or specimens? I think she's probably referring to what's in the statewide biobank.
Statewide biobank. Look, that's a very good question. I mean, we collect samples according to the protocols that are provided by our research partners. So essentially every project involves a negotiation of a standard, you know, a sort of a standard operating procedure that will apply to that study. So that is one of the complex aspects of a biobank such as ours, supporting many different kinds of researchers. In terms of how long samples need to be collected, that tends to be a study specific question. Some samples gain in value over time. You know, but in some cases where, for example, clinical care is evolving rapidly, the research lifetime of a sample might be reduced. So it really depends upon the kind of the physical characteristics of the sample, the downstream analytical requirements, but also the speed at which research is moving within that field.
Great, Thank you. And perfect timing. We're right on the hour.
Excellent. That's great.
So if there are no more questions, we'll move on to our next speaker in this session, who is Dr. Ahn. Dr. Ahn is an Executive Director of Resources and Innovation Cooperative, or RNI, in Korea, where she is involved in projects on biological standards for in vivo diagnostic medical devices and in policy development studies. Before joining RNI in 2019, she was the head of the Department of Education and Research at the Korean National Resource Center, the KNRRC, and responsible for training activities and a number of international cooperation projects related to biological resources and biobanking. Prior to KNRRC, she conducted research on Alzheimer's diseases at the Korean National Institute of Health and managed a biobank of elderly cohorts. Since 2011, she has been the secretary of the ANRRC, which we thank her very much for doing. It's a thankless task, but she does it brilliantly. So as part of that role, she's obviously also a member of the executive. So she completed her undergraduate studies and master's degree at Seoul National University and obtained a PhD in biochemistry from the University of Wisconsin-Madison in the USA. The title of her talk today is Virus Pathogen Resources, Management and Support for Infectious Disease Research. So look forward very much to listening to Kyung-suk. Thank you.
Jane, thank you for the introduction. We put up where is my for some reason my screen got really small. Can you see my presentation?
See the your screenshot, so we've got all the slides down the side as well.
What's happening?
Kwon-suk, would you like us to share the slide for you?
Yes, please. But you don't have my slide.
Hold on, you have to turn your slide off.
I shared my slide, but for some reason the whole screen went small.
Oh, what, Kwon-suk? We don't have your slide.
I know,
No.
but let me
dear.
share my.
Okay. Okay, you got it. Oh no.
Oh, what is it?
Can you scroll down to the next one just to see if we can see?
Can you try full screen?
I don't know how to do it. Screens. Oops. I don't know what I is going to.
Kwon-suk,
Yes.
would you like to maybe leave? Okay, maybe send those your slide to us now.
Oh, I'm sorry. I'll send you. But it worked when we checked.
How about you try the full screen below? You see the tab below?
Should we move to the next session and
Yes.
Yeah,
Dr. Ang
why
can
don't you
email them
have done
across
it?
to you so
I'm
that you can,
sorry.
yeah, that's okay. We'll come back to you.
Yeah. Okay, so maybe shall I go to the next one?
Session, animal.
Session 5? Yes.
Yeah,
Okay.
okay, we'll come back.
So, Dr. Yoshiki, are you ready though? Okay, you're there. Okay, I will introduce you. Okay, so
Thank you very much.
the next session, animal research resources, forefront and diversity, will be run by Dr. Atsushi Yoshiki, who also will be the first speaker of the session. So I will do the introduction for him. So Dr. Yoshiki is a coordinator from Riken Bioresource Research Center, BRC, and the head of Experimental Animal Division, which has contributed for life science through collection, preservation, quality control, and distribution of mouse resources, mainly developed in Japan. He also serves as a member and vice chair of International Communication Committee of Japanese Association for Laboratory Animal Science and a member of the ICLAS Laboratory Animal Quality Network. So Dr. Yoshiki, the stage is yours.
So thank you very much, Lily, and thank you very much for inviting this wonderful meeting. I'd like to say thank you to all organizers. Let me share my slide.
Yes, we can see your slide, but can we have the full screen, please? Okay, great. That's
Okay,
okay.
so let me start session 5 of Animal Research Resources, Forefront and Diversity, with many thanks to organizers for providing us such a wonderful meeting opportunity. My name is Atsushi Yoshiki from Riken Bioresource Research Center, Tsukuba, Japan. So I'd like to chair this session. I hope every participant is encouraged with a meeting titled Beyond COVID-19 and Sustainable Research Resources. Almost 2 years of global suffering from the COVID-19 pandemic, we should remind ourselves the importance of sharing knowledge, technologies, and bioresources, and advancement at the cutting edge. We really appreciate this timely and helpful opportunity by the organizers to share updated information of animal research resources forefront and diversity by 2 speakers from Japan and South Korea. So the first speaker, myself, and we would like to talk about mouse resources, so-called the premier model organism for the human. And the second speaker, Dr. Jae-won Hu from National Primate Research Center, CREEP, will talk about an advanced primate research base in South Korea. So let me start with my slides. So the title is Advancing Mouse Resources for Studies, Health and Disease. So recently, more emphasis of data and phenotyping and genome sequencing
And this slide shows the image of our Tsukuba campus. So RIKEN Bioresource Research Center was established in 2001 as a comprehensive biological resource center in Japan. Currently, can you see the pointer? Currently, RIKEN BRC consists of 7 infrastructure divisions and 6 research development teams under the director Toshihiko Shiroishi and senior advisor and a former director Yuichi Obata. Regarding the major animal facility in the campus, we use 3 facilities: quarantine and rederivation and a barrier facility. We also closely collaborate with Bioresource Engineering Division for cryopreservation, the mouse phenotyping team for comprehensive phenotyping for human disease, and integrated Bioresource Information Division for dissemination of web catalog and genome sequence information. The experimental animal division of RIKEN BRC has been designated as a core facility of National Bioresource Project, so-called Nbrp, by the Ministry of Education, Culture, Sports, Science and Technology. Our program is called the covers 30 divergent model organisms. The National Infrastructure Project has been supported by the Japanese research community and the government, therefore continued as long as for 20 years. Our mission is in this program is to collect models for human diseases and gene function mainly developed in Japan, preserve variable mouse strains for long term, and quality control for animal health and genetic integrity to ensure reproducible experiments, and distribute high-quality mouse resources to the global scientific community. This table shows our achievement statistics for the last 5 years. The biggest mouse resource archive in Japan and distribute over 2,500 mouse resources to Japanese and overseas scientists every year. During April to May 2020, last year, we suffered from spread of COVID-19 and some depositions, and the distributions were suspended or postponed until partial resumption of the activity in late May 2020. Thereafter, we suffered for several months for difficulties to ship mice to overseas due to reduction of the air transport. As an international hub of mouse resources, we distribute our mice to international scientific community. To connect globally, we participate in several international networks and work together other mouse repositories around the world. RIKEN BRC has participated in the International Mouse Strain Resource, IMSR, a one-stop shop database operated by the Jackson Laboratory USA to register mouse resources developed by Japanese scientists and share those resources with global biomedical community. The IMSR list can direct scientists to the holder of repository with one click. This function has increased the visibility and accessibility of our mouse resources. ANRRRC is an Asian Australian network of mouse resources for communications and technical exchanges in the region. we have participated in the International Mouse Phenotyping Consortium and contributed to the production, preservation, and quality control phenotyping and the distribution of knockout mouse resources. And this GMMCC is a global mouse models for COVID-19 consortium to post our own biological resources useful for COVID-19 studies. So this is our web page, and phenotypic data for each strain is an important key to evaluating the strain as a potential human disease model or tool to study disease mechanism. The Integrated Bioresource Information Division has recently upgraded the mouse web catalog by manually adding 24,000 phenotype annotations to over 4,300 strains from publications, so that users can find a specific mouse strain based on phenotype ontology terms in addition to gene names. This increases findability of the strains. We have our homepage proactive outreach corners focusing on specific mouse strains, such as today's tools for functional analysis and today's model for human disease, and mouse of the month to provide researchers with information about advanced strains and facilitate the use of such strains for cutting-edge research. We collaborate with the National Bioresource Information Center and other programs such as Genome Information Upgrading Program, and this program, Fundamental Technology Upgrading Program. The purpose of these programs is to enrich and expand genome information of biological resources in the core facilities and develop novel technologies and tools to create much advanced models. Recently, we collaborated with Genome Information Upgrading Program with long-read sequencing of the genome in Japanese strains. So this is the web page, the Nbrp Genome Information Upgrading Program. So collaborations with the program that contributed to the addition of genome sequence data of wild-derived strains of Mus musculus, MSM, and JF1 in the MOG Plus mouse genomic variation database. Such genome information is essential and useful to advance mouse resources with sophisticated genetic modification by genome editing and standardized phenotypic data. Our divisions and teams are collaborating with external clinical experts and creating much advanced mouse resources, such precision mouse models for human diseases with patients, mutations, and variants. We have built up a production pipeline for knockout and knock-in mice by genome editing, and comprehensive mouse phenotyping pipeline, so-called Japan Mouse Clinic, to expand our molecular understanding and knowledge of health and disease in digital form to optimize use of such knowledge. Well, thank you very much, all people listed here, for many advice, support, and excellent works. And we hope we can meet directly face-to-face, but with an appropriate interval in near future, and freely travel to each other around Asia. Wish you safe and healthy. Thank you very much. I'm happy to take questions. Well, stop sharing. Yeah, thank you very much. So do I have time for question? So I do not have any question in Q&A box. So may I move on to the second speaker?
Yes.
Yes, so I would like to move to the next speaker, Jae-won Hu from Laboratory of Molecular Genetics, National Primate Research Center, KRIP, South Korea. Briefly, he has appointed as Associate Director of National Primate Research Center, KRIP, and Principal Researcher of the center since 2018, and also Professor of University of Science and Technology since 2019. Dr. Hu has obtained his PhD of Biology at Pusan National University, South Korea. His major interests are comparative gene analysis between human and chimpanzee, gene domestication mechanism of retroelements such as lines, signs, hubs during the primate evolution. So we'd like to listen to his talk about primate research in Korea. So Dr. Hu, can you start?
Yeah, I can start. Can I share my
Yes, we can see your slide.
screen? Okay.
Could you turn to full screen? Yes.
Yeah.
Yeah, thank you.
For my presentation, thank you to the organizing.
Yeah, your voice is
Small?
maybe, yes, small. So
Is it good? Is it okay? Can you hear me?
could you,
Small?
yes, small.
Oh, okay. Can you hear my voice?
Yes. Please try
Okay.
louder.
I make louder. Yeah. Before my presentation, thank you to the organizing team for inviting me to the ANRRC conference. Today's my talk's title is Introduction of National Primate Research Center in Korea. This picture shows the total aspect of our center. Can you this one? Yeah. This building, I work here. Now I am in this building. Now I started. This slide shows the current situation of primate resources in Korea from KFDA record. Upper figure, this figure, shows the current status of monkey import, and this down figure shows the usage number of monkeys in biomedical research. This yellow line indicates the amount of monkeys used in biomedical research in Korea, and this green line indicates the important number of experimental monkeys in Korea. Maybe this year and last year, the record of usage number of experimental monkeys will be remarkably increased. Because due to the COVID-19 pandemic, we will use many monkeys for experiments. And this is a representative institute in Korea, having a housing facility for primate. Red color, this one. Red color is industry area. only one company, Orient Bio is a Korean only one company, have a monkey breeding farm in the other country, specifically in Cambodia. And this is a purple color is the industry area. Purple color of Seoul National University is blue color and green color is a government affiliated institute. This is a KIT for toxicology. This is a KBio, Health and Experimental Animal Centers. Our center, National Primate Research Center, also included in this area, a government affiliated institute. All of these institutes have a monkey housing facility and provided a monkey experiment service. During the COVID-19 pandemic period, primate resources have become very valuable resources worldwide, and specifically American, China, and Korea also. Because of short supply, the price of monkey have reached more than $10,000 in Korea also. This is an international monkey shortage crisis. And also animal rights groups campaign in relation with monkey transport using the airline cargo system. Stop Transport the Monkey campaign is one of the old threats for monkey resources. Nowadays, only one airline, Air France can transport the experimental monkey by cargo. That means all other countries have some trouble to transport the experimental monkey from one country to another country. This is our center's mission to establish the national primate research base to be an infrastructure of nationwide to improve public health and establish a bioeconomy. One of them is to secure, maintain, and standardize SPF primate resources. And second one is to construct preclinical infrastructure for the development of regenerative medicine, bioorgans, and new biodrugs. And third one is to support research solving national social issues such as COVID-19. Another one is to establish primate research infrastructure to support the research on disease and novel drug development. And last one is to develop research materials derived from SPF primates. This is our history. First time we started as a one paper. The paper name is National Research Project on Non-Human Primate Research. In 1998, we started. After that, 1 year later, 1999, first introduction of SPF monkeys in Korea for biomedical research. And 2025, we completed National Primate Research Construction in Ochang, these centers. And 2016, we divided this National Primate Research Center into two different centers. One is NPRC, another one FARRC. That means NPRC is focused on the primate and FARRC is focused on the miniature pig. Next year, we divided into three centers. One is NPRC for primate experiment, and PRC make a monkey breeding colony in Korea, and they make a monkey breeding colony. And the PRC is focused on the miniature pig. This is our location. Our National Primate Research Center is located in Ochang. The Ochang is located in the center of our country. Then this kind of geographical advantage, all other Seoul and Busan, Gwangju can access very easily in Ochang. Therefore, we established our primate research center in this area. And this building is Beijing Research Building, and this building is a quarantine building, and these two, another building is a primate experiment building, and this building is a futuristic animal resource building. However, these four different buildings have small cases of a monkey housing facility for monkey experiment. And another location of a PRC is in here, like this. Jeongup Center is a more southern area compared to the Ochang. because monkey breeding colony center should be located in the more warmer place than our center. We selected these areas. This one is a basic research building. This one is a quarantine building. All other buildings are primate housing building. And this small circle is a primate castle. And this is our organization. I'm director of these centers. We have 3 different divisions. First one is research support division. Second one is resource management and standardization division. Another one is research groups. And resource management and standardization division, we have a veterinary, medical team, and breeding team, and then health monitoring. We have 3 different teams. And we have a clinical pathology group, and the molecular genetics group is my group, and another one is a neuroscience group, and the last one is immunology and infection disease group. Nowadays, we made another 2 new groups. One is genome engineering laboratory, and another one is regenerative medicine laboratory. And this is for research support field. As an institution focused on primate research in Korea, we will grow together with industry, academia, and the research institute. That means we are, as a national infrastructure of primate research, we provide all over field in response to the demand of industry, academia, and research, because we are national infrastructure for primate research. This is the record of our research support. During the 6 years, we provided more than 5,000 cases support. Among them, more than 20,000 resources primate research. We provided monkey experiment to our primate resources. Another one is mainly material. Among the material, most frequently used material is the blood samples. And this slide shows the current status of the year by year. We can use our records. During the 6 years, the trend could be increased slightly. And this is our resources, NPRC and FARRC. Nowadays, we have more than 250 heads of monkeys, specifically rhesus monkeys, and cynomolgus monkey, and small number of African green monkeys. And we have a SPEP miniature pig, more than 100 heads of miniature pig, and we make animal-derived resources, for example, the nucleic acid and cells, specifically primary cells, and tissues, and the genetic database for the monkey. This resource is in PRC. In the case of PRC, we have more than 800 heads. This monkey's number is decreased very fastly because of the COVID-19. We cannot import of the monkey. Only we used this kind of monkeys. Therefore, the number is only 800 monkeys. And this system is totally different from the Ochang centers because PRC is the open cage system. And this is a housing facility. Animal facilities based on national standard, specifically NH guideline, and we consider the animal welfare. Therefore, we made very different toys, and we used the mirrors and fresh fruits for the monkey welfares like this. And we make the SOP, species-specific SOP, annually and develop this SOP, Standard Operation Procedure Manual. This is our facility and instrument. Advanced research instrument, for example, the 3D MRI and angiography. This one is a PET-CT. All of these systems, angiography, 3D MRI, and PET-CT is for human. And another micro PET-CT is only for the rodent. However, in the case of the monkey, all of this system is compatible with the human system. We use these human machines. And this is a micro-manipulator system. We provide one-stop research system, experiment agent service, our slogan is from housing to experiment. Another our facility instrument is Animal Biosafety Level 3 facility. From 2015, we established this facility, and nowadays we provided the COVID-19 experiment, we provided that related system. After the COVID-19 pandemic, we made the disease model of COVID-19 using the cynomolgus monkeys. This paper showed the world first model in the world, and from this model, we provided experimental service more than 10 companies. This is our main research area. Our main research area is development of brain disease model using primate. Specifically, we tried to make a degenerative brain disease. Nowadays, we focused on 3 different disease models. First one is stroke, another one is Alzheimer's disease, last one is Parkinson's disease. In the case of the stroke disease, we make them surgical disease model, and another Alzheimer's disease and Parkinson's disease model is the chemical introduction model. In the stroke model, we made them middle cerebral artery occlusion method, and Alzheimer's disease model, we use intracerebral ventricular injection of streptozotocin, and last one, the Parkinson's disease model case, we made intramuscular injection of MPTP. And all of these brain disease models evaluation was conducted using the imaging device, 3D MRI and PET-CT. And this is another system. After the disease modeling, we compared and analyzed these brain samples, MOTEM samples. This system is MR-compatible brain tissue cutting techniques. We established this kind of system. That means this picture is the tissue pictures, and this one is MRI imaging. We made this one and this one is compatible and analyzed the same resolution. And another system is MRI stereotoxic system, because in the case of monkey, we should inject some kind of small amount of materials exactly, repeatedly in the same position and same region of the brain. First time we use only stereotoxic system. However, in the case of the monkey's brain, only for the stereotoxic system cannot make repeatedly same injection. Therefore, we developed the MR stereotoxic system. This means in this MR stereotoxic system, using the MRI, we can inject the totally same site and same amount and same depth using the MRI system like this. And another our main research area is transgenesis system in monkey and miniature pig. For generation genetic disease model and bioorgans, we use 2 different transgenesis system. In the case of monkey, nowadays we used virus-mediated transgenesis system. In the case of the miniature pig, we can apply the somatic cell nuclear transfer system. And this is our main research area. After make a disease model and some kind of a surgical model, we try to assess the behavioral assessment method, because in the case of monkey, if we have some question about this monkey's brain, we cannot open the monkey's brain. We commonly use the non-invasive assessment system. Therefore, we established the video image-based activity measurement like this. All of the monkey's activity was recorded this kinetic camera, and then we analyzed this kind of the monkey's image data. And this one is also first time we tried this kind of the neck color system, and this device was adopted in there. However, this is so heavy, we made another advanced version. And this one is a cognitive test using touch screen computer system. And this is a delayed match to sample test DMST experiment using the monkey. this test is assess the cognitive ability like this. Okay, I'm sorry. This is another behavior assessment method like this. First time we can record the Kinect camera and like this, and all of the video image was recorded. First time we calculated all of the monkeys activity like this, sitting, stand, and rotate. We count one by one by hand. However, more time-consuming method. Therefore, we analyze like this using automatical method. How can we analyze them? And we developed this kind of automatical assessment system like this. This is a monkey behavior analyze method like this. Some monkey like this food, another monkeys like this. Therefore, we can analyze the eating behavior like this. First time monkey was started like this, and this experiment also check the monkey's activity. He met on new materials like this. How can have their behavior like this? And this is a guide assessment system. From this system, we can analyze the monkeys' walking system and the monkeys, how they walk in the guide tunnels like this. This is a monkey's footprint like this. And we established the behavior assessment method. This is hand preference test. Some kind of monkey use right-handed, another monkeys have left-handed, and we can discriminate using this system. And in this experiment, the hand dominance test. Oh, this is a finger maze test. This is this test for the cognitive test like this. This is a failure. This food was dropped in this area to this one. He cannot eat. However, like this, he can handle this food very easily like this, and then he can get this big sized cracker like this. Success. And lastly, yeah, his main research area is a long-term project using the African green monkey. First time in National Private Research Center, we import the African green monkeys from the Japan. However, after then, their age is more than 20. We cannot use any other experiment. Therefore, we try to analyze 1 year by 1 year, because in the case of monkeys, their environment is well controlled environment. Therefore, we analyze the aging, the naturally aged monkeys system. We can analyze using this RNA-seq data and material-seq data and double-JARS data. We collected all of them, 1 year to 1 year, more than 5 years, analyzed them, and we published the longitudinal profiling of blood transcriptome in African green monkey aging model. And nowadays, this aging model was followed the cynomolgus monkey. We tried another 20 aging groups, and then nowadays we collected this kind of different samples from the blood and swab and sperm and analyze annually. Thank you.
Thank you very much for comprehensive explanation of primate research in Korea. So we have a Q&A box question from Jennifer. Can you unmute the microphone and talk?
Oh, thank you. Yes, my question was about clearly animal welfare is very important, and you mentioned the providing monkeys with like fresh fruit and toys and mirrors and things like that. I'm aware, I mean, that the differences in those kinds of enriched environments can lead to reproducibility issues between facilities. I mean, that's true for mice. I'm sure it's much more pronounced for monkeys that are much more intelligent. So I'm wondering if you could comment on how you standardize enriched environments for the monkeys. Is there a worldwide effort for that?
Yeah. In the case of the monkeys research area, the most important part is animal welfare. You can see first time our facility was by like this system. First time we housed the monkeys, this kind of the one cage, one monkeys. Although we make a more bigger cage, however, we did not have a housing cage. That means the monkeys should be housed in family. However, first time we could not do. Nowadays, we tried to make another big cage, and some monkeys could be relocated to the family. Nowadays, we transferred. However, the change is very slow because the monkey facility. That means more funding, more money was needed. Therefore, we very slowly we changed our facility from this kind of the cage housing system to the big sized family cage system. And this is our what nowadays our goal to make all of our monkey was relocated to the big cages with a family housing system.
Thank you.
Thank you, Jennifer, and thank you for answering Jaewon. So it's a time now to move or back to the session 4. So I'd like to close the this session. Thank you, Professor.
Thank you very much, Professor Yoshiki, for sharing the session. And before we go to the last session, we have to go back to Jane and Kwon-suk again for the previous session. Please, can you maybe make it short as well? Thank you.
Thanks, Lily. So we've already introduced Dr. On. So Kyung-suk, if you could try your slides again, please. Thank you.
Please.
No, we will share the slide for you.
Oh, sorry. Yep.
Okay. Thank you for the introduction and sorry for the delay. I'm going to talk about how virus pathogen resources are managed in Korea. Next. So first, regulations, and I'll introduce the several biobanks and also talk about safety considerations. Next. Well, Jenny really gave a very nice overview about the human health. And since this COVID-19 pandemic, the importance of biobanks are, especially the pathogen biobanks, are getting more attention for the research and development on infectious diseases. Next. Next. Well, this is the master plans and legislations on bioresources in Korea. In 2009, the Biological Resources Act was enacted, and this gives the basis for all this bioresource management plans at the national level. In 2016, the Pathogen Resources Act was enacted. Next slide. So in this, well, there are other laws about the pathogens pathogen infectious disease preventions, but the government recognized the importance of these pathogen resources, and this act defines the pathogens as following pathogenic organisms, bacteria, fungi, viruses, and also the derivative materials. And this law defines the responsibilities of the state and establishment and operation of national culture collections, and also specialized pathogen resources bank. Next. And this act, no, before, so this act tells to people to deposit the pathogen resources at the National Culture Collection for Pathogens, NCCP, or specialized bank, or biological resource centers. And this is to support the research and development, and also the health industry in Korea. Next slide. And I will introduce several biobanks. Next slide. NCCP, this is the main building of the NCCP, and they collect and accept depositions of pathogen resources. And they also develop pathogen resources. And also they understand, they have the central role of managing the pathogen resources in Korea. Next slide. They also do the conducting research on technologies and bioinformatics too, and also chairs the Council of Microorganism Research Centers of the many different ministries, like Ministry of Environment, Ministry of Ocean, Ministry of Agriculture, and Ministry of Science and Technology. Next slide shows the statistics of collection and registration of microbial resources between 2017 to 2019. In 2019, they provided resources to 174 institutions in Korea. Distribution to private sector is increasing. Jenny talked about the underuse, and it is a common challenge in Korea too. And they did not publish the 2020 statistics yet, but I expect the numbers have increased. NCCP handles microorganisms, bacteria, and fungus, and virus resources. And the following 3 biobanks are specialized in virus resources. Next. So this is the Korea Bank for Pathogenic Viruses. There are 6 specialized banks in NCCP network, and this is one of them. And this bank used to be the part of the ANRRC bank, started in 2005, and now it is supported by NIH. This bank is at Korea University and has various virus pathogen resources, nucleic acid, anti-sera, and has biosafety level 3 laboratories. And next, the Human Serum Bank. This one has high-risk viral antigens and antibodies, such as human hepatitis viruses and human immunodeficiency. And this is supported by NRF since 2010. since some of the like HIV, we have very low incidence, and they collaborate with African countries and Asian countries. And Professor Cha and colleagues established a network of laboratory medicine specialists from 14 Asian countries. ANRRC, which is the Asian Network of Clinical Laboratory Standardization and Harmonizations. This bank used to be at Chung-Ang University Hospital, but now the bank is at Institute Pasteur Korea. Next. The next bank is this co-seed. Sorry, go back. This co-seed is a network that deals with high-risk pathogen resources. This collaborations talks with many countries around the world. It is at Chungnam National University, and it moved from actually Busan University. So the previous three biobanks, I think it is encouraging that these three pathogen biobanks continue to be in operation through changes of funding and changes of physical location in the institutions. And next, I'll talk about the safety issues. Next. I think you all know that there are 4 levels of biosafety, and level 1 and 2 are basic levels, which is most labs handling microorganisms belong to. And level 3 and 4 are high risk level, which requires containment, such as controlled access and airflow and personal protections. Next. So KCDC's direction for COVID-19 samples are like this. SL2 for inactivated specimens and BSL3 for viral cultures, etc. And these high level 3 and 4 are subject to permission and inspection by KCDA. It used to be CDC, and it was upgraded as an agency during this COVID-19 crisis. So as of January 20 this year, there are 75 SL3 laboratories are in operation in Korea. And some facilities are open to outside researchers on project basis. And Institute Pasteur Institute is building additional BSL laboratories to provide an access by our researchers. I was involved in the project for strategy development on jointly utilizing virus core research facility. And I'm glad that the new project will start in the next year to researches on virus pathogens. So the biobanks I showed in previous slides all have BSL3 laboratories. Next. Well, there is another consideration. The pathogen resources are genetic resources defined in Nagoya Protocol. So the exchange of pathogen resources between countries is very limited because of safety issues, and also each country have different regulations. So in Korea, under the ABS Act of Korea, you have to report I get permission from the Ministry of Health and Welfare to export or import pathogen resources. So here showing the comparison of the two laws. So next slide is my last one. To summarize that Korea has the separate law for pathogen resources, and the purpose of this act is to enhance the public health and to contribute to the development of the healthcare industry, and the contribute to the national economy by promoting collection, management, and utilization of pathogen resources. And the human there are other laws that of health biobanks, which is Bioethics and Safety Act, and also biosafety is the regulation by LMO laws in Korea. So to summarize, that handling pathogenic samples requires to follow safety precautions, and all those have to meet the requirements. And export and import of pathogen resources regulated under the ABS law too. This is my talk. Next slide is the last one. Stay safe and healthy. Thank you for your attention.
Hey, Dr. Arnold. There are a few questions in the Q&A, but can I just ask you, do you collect clinical information on the donors to follow up for particular, who've had particular viruses or bacteria?
Yes, but when Biobank provides to researchers, it is de-identified. So
Yeah,
they can provide de-identified information. We have a personal information protection law.
Yeah, but you collect long-term clinical outcomes of people who've had the same infection. Yeah,
yes.
Yeah. Okay. There's 3 questions in the Q&A. Can you see those? We probably don't have time for all of them. Actually, leave it up to you which one you want to answer.
Okay.
You might just want to type the answers to the other 2.
Not be collected for the materials and requires substantive the bioavailability with the type of pathogen. All the live pathogens are very hard to handle. So it is only by very specialized people and biobanks. And so the BSL-3 laboratories are for these live viruses. And they have to follow all these precautions and regulations about the shipping, packaging, and everything. So you have to follow all those laws. We have a infectious disease prevention law, which tells how all these things you have to follow.
Yep.
So do you check the virulence properties time to time? Well, the Korean FDA provides the national standard reference materials. And in that case, they do check the stability. I am involved in one of those projects. And as they do, in the NCCP, of course, they do check the viability.
Yep.
And thank you very much.
The last one is there a bank of closed genes of viruses?
What is it?
Is there any bank of cloned genes from viruses?
Cloned genome virus. Yes, of course, the derivatives include some DNA and RNA materials, and also the anti-sera and other resources.
Okay, I think in the interest of time, Lily's waiting. Thank you
Yes.
to both the speakers in the human session.
Yeah,
Thank
thank you
you.
very much, both
Thank
Jane and
you.
Kwon-suk. I know that there are a lot of questions there, but yeah, because of the time, I have to move to the next session, which is the last one. Thank you. And so the last session is the law and regulation will be shared by Dr. Shyam Kumar Sharma. And Dr. Sharma has a vast experience working in the area of genetics, crop improvement, biotechnology, and plant genetics resource management. And he has won several national and international fellowships and awards during his career, spanning over 40 years. And these include the Commonwealth Academic Staff Fellowship and also Marie Curie European Commission Fellowship. And of course, he is also the board member of the ANRRC. So please welcome Dr. Sharma. And sorry, I have to say that maybe you have to be a little bit brief and stick to the time. Thank you.
Okay, thank you, Lily. I also welcome all the board members and the participants to the session 6. And this session was also introduced by Philip in one of the earlier sessions where he mentioned about ABS and DSI. And as you are aware that the countries in the Asian region are at different stages of developing guidelines on access and benefit sharing. Besides, the guidelines are being developed are also of variable nature as these are based on their domestic laws on bioresources. ABS in these countries are governed by multiple statutes handled by multiple ministries, issuing multiple guidelines and executed by the multiple agencies. And therefore, there is a need to standardize and harmonize the modalities for access to genetic resources before ABS provisions are implemented. Similarly, the DSI is another very important issue which is being debated, and there is a need that all the countries in the Asia particularly, they must come on the same platform when issues on ABS as well as the DSI are being debated, so that in course of time we are able to make use of the bioresources in the Asian region. With this small background, there are two speakers in the present session. The first is the Sunil Archik. Sunil Archik is working as a principal scientist and ICR National Fellow at the National Bureau of Plant Genetic Resources, New Delhi. He has a very wide experience and was instrumental in establishing National Genomic Resource Repository. Dr. Archik has established a PGR information facility at NBPGR, and he is also a member of teams which has organized many national and international seminars. He has represented India in the treaty governing body meetings. He is also a member of the Scientific Advisory Committee on Global Information System, as well as the working group to enhance the functioning of the MLS of the treaty. He has also represented FAO, CGR, FA in these meetings. With this small introduction, I would request Sunil Archik to make a brief presentation. Sunil Archik.
Thank you very much, Professor Sharma, for that kind introduction. I hope I'm audible clearly and my slides are visible.
Yes, little louder, please.
Yeah, sure. Thank you very much for the organizers, to the organizers for giving me this opportunity. Thank you, Toshiko-san. I'm going to speak to you on accessing genetic and genomic resources in India, vista of regulatory framework and opportunities. And I'm not going to tell you a lot of quantitative data on that, but tell you principles on which we actually function here. I am from Indian Council of Agriculture Research in India, where the bioresources availability, the conservation management of bioresources related to agriculture is managed by 5 distinct bureaus. National Bureau of Plant Genetic Resources, this is where I come from. And we also have national bureaus on animal genetic resources, fish genetic resources, microbial genetic resources, and insect genetic resources. In this talk, I'm going to specifically focus on plant genetic resources. So what plant genetic resources are accessible for people within and outside India? We have IP protected varieties. We have gene bank accessions conserved in huge gene bank. And these gene bank accessions are, some of them are registered germplasm for specific traits. And we also have those germplasm lines accessions that are designated in the multilateral system. We also have material in the private domain. And then we have huge amount of diversity existing in nature, in the wild. We also have DNA available in different institutions. So we have to talk about that. When we see at that, considering the amount of diversity that is present in this hotspot, the world, India, we have in the gene bank around 400,000 accessions in the form of seeds across various crop species. We have 2,000 accessions in the form of tissue culture repository in vitro conservation, about 12,000 accessions as a cryobank, and around 9,000 accessions specific to plants. We also have around 26,000 accessions designated in the MLS multilateral system, and a little bit of accessions are designated as registered germplasm. And then some of them, which are a little bit common with the gene bank as well as outside, are plant varieties that are registered. That means these are the ones which have got plant builders' rights as well as farmers' rights. I'll tell you about that a little bit. So before we actually understand the vista of regulatory systems in India, we must understand the institutions that are involved. First, 2 institutions which are very important come under Ministry of Agriculture in India. One in that is Protection of Plant Varieties and Farmers' Rights Authority, which is present in New Delhi. This is an authority which implements the plant breeders' rights protection along with farmers' right protection. This is in lieu of UPO kind of system. And this is the only system in the world which implements both farmers' rights as well as plant breeders' rights simultaneously. The second one is National Bureau of Plant Genetic Resources, which is the where the gene bank is hosted, Indian National Gene Bank, one of the 3 large gene banks in the world. We also have a small genomic resources repository. Apart from this, we have National Biodiversity Authority, which is located in Chennai, the southern part of India, which comes under Ministry of Environment, Forest and Climate Change, which is the nodal agency in India for the access to any kind of bioresources. We also have Ministry of Commerce that comes into picture because wherever there is a commercial exploitation, intellectual property applications, they are linked to these access to genetic resources, this particular thing comes into picture. So quickly, one by one, what do we have in these? In the gene bank in India, we have a huge repository. We have around, I just mentioned, more than 400,000 accessions belonging to all major and minor crops. And one can see all the details on NBPGR's website and then application. We also have an in vitro bank where tropical fruits, temperate fruits, tuber crops, bulbous crops, various. You just name any one crop group, it is cultivable in India in one or the other part of India because of the variation in the agroecology is available. And then we also have cryogene bank. We have a genomic resources repository. I must mention here that the chairman of this particular session, Dr. Sharma, was instrumental in establishing this particular repository in India. So we we have about 9,000 genomic resources available. But what way these are going to be accessible and how we are going to have what kind of MTAs are there, what kind of benefit sharing is possible, etc., are still being explored. All the information, when I say all the information that is passport data, trade-specific data, all these information are accessible freely to anybody in the world on the NBPGR informatics systems. You just scan the QR code on the screen, you will go to that particular page. The second organization is Protection of Plant Varieties and Farmers' Right Authority, one which provides plant breeder certificates along with farmers' right certificates. So at this point of time, more than 160 species are provided plant breeders' rights. And the usual system, the same system that is followed globally by UPO or any other organizations, that distinctness, uniformity, and stability are checked, and then plant breeders' rights are allotted. So you can see here, more than 5,000 breeders' applications have been received so far, but more than 10,000 applications have been received by from farmers. So till the COVID-19 struck our system, more than 4,000, say around 4,500 certificates of protection had been issued, out of which 1,800 to farmers, 800 to breeders. And we also have different systems of protecting essentially derived varieties. And the ones with extant varieties, that means the varieties that were already in place before this entire legal system came into place. And this particular authority, National Biodiversity Authority, is very, very important because this is the nodal agency through which all other agencies operate in India for anyone from outside India or even from within India to access bioresources. So NBA regulates biological resources, not just resources, associated knowledge for research. If you want to carry out any kind of biosurvey or bioutilization, or you want to carry out commercial utilization of already existing material, you want to obtain IP rights from the materials that you have already accessed, or you want to transfer the results of research of this the material that you have accessed or the knowledge. So in all these entire realm of bioresource utilization access is managed by National Biodiversity Authority. It also deals with a number of techno-legal issues, and then it follows, like the previous speakers mentioned, that there is a clearinghouse mechanism so that it's taken care. I'll quickly go to the next one. Before we actually talk about how things are different and why things are different in different countries with respect to the access to genetic resources or even genomic resources, same breath, we must understand that we inherit some legacy. Okay, India is extremely gene rich. It is primary and secondary center of origin for multiple crop species. So historically, India was not seriously dependent upon outside world for specific crops or crop genetic resources. I'm telling much before the systematic breeding started. So the thinking is different. The second one is, we have diversity, we have extreme diversity. We have all possible agroecologies and the food systems present from north to south, and it's a huge country. So experimentation is possible. The material from one part of the India to introducing to the other part of India itself is a Herculean task. So in itself is a complete system. We have diversity. Yes, we have extreme diversity in cultural diversity. We have curative diversity. We have culinary diversity. That means it's almost a self-contained system all along. So it's not that easy that somebody can from outside just quickly come and access material. So it was not that easy. Of course, we have signed all these international agreements and treaties. Yes, things are in place now. Slowly and slowly, things are being opened up. What is very important now, however, to understand is the breeding, plant breeding has been in the public sector traditionally in India. It was not dependent upon private sector. So which is why the access and the utilization was linked in a self-contained system. That means it was not different. Somebody is accessing from outside and therefore utilization has to be regulated or commercially manipulated, etc. It was not like that. The last point in this is about the ownership. How do we manage the ownership, which are split in different kinds of ways? Sovereign ownership, community ownership, individual ownership. It is not very easy. As I just mentioned, a specific crop species diversity in the form of land races is distributed in India all along. For example, on the left, you can see Oryza sativa, the indica sativa rice. On the right, you can see various Vigna species. So it is very difficult to identify the provenances or the small regions to ensure that, okay, we can enter with now in an AGOA protocol, we can start implementing this. So it is not easy. Okay, what is the nature of access? So nature of access is not uniform or single. Nature of access depends upon who is accessing, who is trying to access the material, and what is the exact material, what is the source of the material. So it can be a restricted access, it can be facilitated, or it can be free access. For example, if private sector is involved, particularly when the shares are owned by outside the country, then there can be a restricted access. There are different laws coming into picture. If it is public sector researcher and students are there, then it is facilitated access. There are MTAs which are there. And if it is the access is by the farmers or the community, then it is free access, because the gene bank is actually the custodian of the germplasm which we have collected from them. Okay, now all of you specifically know that access is inherently linked to benefit sharing. So whether the access is for specifically only R&D, and then after that it is in the public domain, the product comes into the public domain, or the knowledge comes into public domain, it is fine. Nobody is questioning. But then once if it goes for commercialization stage, yes, then one has to talk about benefit sharing. Now, whether it goes to commercialization stage directly or through some kind of protection, intellectual properties, then again, different laws and different the forms and different, let's say, additional steps come into picture. Now, what happens? How exactly the benefit sharing is implemented? All case by case basis, because there are different multiple channels. What we have to understand is plant genetic resources in India and many other developing countries or gene-rich countries is not just economic resources. There are some countries' pride and then political sensitivity, many other things are involved. The second one is because so many institutions are involved, so many laws are there, because we are members, we are signatories of Convention on Biological Diversity, for the treaty, or various other international, and they are all implemented through different agencies. We require lots of harmony across laws and institutions. There are challenges of scientific and legal definitions when things go to court of law. And then it requires cases. That means we require precedents of solving quickly the things. So all these are required, and there are some stray cases of what we call as exceptional, let's say, misuse of IPR. Okay? So these are all required. Now quickly, there are two ways of looking at this. Accessing plant genetic resources from India. I'm not saying about of India, from India, which is present right now here. Now, people who argue that everything is all is well, all is set, they say that entire passport information of whatever we have in public domain in gene bank is available to free access. All the trait information is also available. People are improving, adding to all that. So information is available. The moment information is available, the access starts. The information access is the prerequisite for material access. And we have shared material in a multilateral system. So India also has rules and procedures in place for various ways of accessing material. We also have access open to private and public. So what is the problem? So other side of the story is why we need an improvement in this particular system. So when we have all this data of the material that we have in a gene bank, do we have all these georeferenced? Do we have all these material genotyped by, let us say, SNPs or SSRs or whatever technologies that are available? So these are the questions. Improvement, there is always room for improvement. Another part is whether we have shared everything that is supposed to be shared in MLS. No, not yet. So this also will improve. People say that procedures are complicated and there is a delay in processing their requests in Biodiversity Authority, but they have improved tremendously. Now things are all online. The forms have become very short and small. People can register their companies and then need not fill everything again and again. So there are many things which are taking place. But one small thing which I have to just put in this context is whether the material that is originated from India is available from India, or whether it is available from anywhere else, doesn't matter as long as the purpose is solved. For example, just look at this graph. This just says that global distribution of Indian origin germplasm conserved by various gene banks. So you can just see here that see around 35% of the ICRISAT gene bank is from India. Around 16%, 17% of rice in IRI is from India. And similarly, you can see here, whatever is in case of pigeon pea, in case of Setaria, in case of amaranth, all these very important food and nutrition crops, which ensure nutritional security in the world. You can see that Indian origin accessions dominate in most of the places. That means this is in the Svalbard seed world. Okay, now just to summarize and then quickly tell you how, if any one of you is interested to access material from India, what are all the different ways? There are 4 distinct categories. Number 1, if the germplasm is not covered under any collaborative research project, that means you and I are not having any kind of a collaboration in kind of research, but you want to access something, then you have to come through National Biodiversity Authority, and there are very specific sections and then specific forms online system is present. Let me jump to category 3. We have Annex 1 crops, which is part of International Treaty on Plant Genetic Resources for Food and Agriculture. In this case, National Biodiversity Authority has delegated that particular process to ICR, National Bureau of Plant Genetic Resources. So we manage that one from the National Bureau of Plant Genetic Resources. It's from the Annex 1 crops of the MLS. Category 4. If it is Indian researcher, government institutions want to carry material from India to outside for research, or in case of emergency, for example, there is a rust disease devastating, there is some other diseases devastating quickly. Let's say we want to test all the material in Africa, we want to test in Australia, we want to work with some other countries, they come through category. I want to invite your attention to what is category 2 and which is very attractive from my point of view. That is germplasm under collaborative research projects and work plans. This supports 2 things. This facilitates 2 things. 1, that you and I can work together on the common causes. Number 2, it allows me to share germplasm very freely, and there are very easy way of manipulating the, when I say manipulating, taking care of the commercialization. And so the most important way, the most beneficial way of sharing germplasm from India is by collaborating with one or the other Indian research agency. And all the details are anyway available online. I just stop it here and then thank all my colleagues. As you all know, any kind of bioresources work is not an individual work. It's a team work, the current team and the past team, and all other funding agencies. Thank you very much. Thanks for your attention.
Thank you, Dr. Sunil Archik. Now the second speaker is Dr. Suzuki.
Yes.
Dr. Suzuki is the Director of the Intellectual Property Unit and the National Institute of Genetics Innovation at the NIG in Japan, which is a member of the research organization of Information and Systems at the Inter-University Institute in Japan. Dr. Suzuki has been participating in CBD-related conferences over the last 10 years and is one of the Japanese delegate as a scientific advisor. He was also a scientific advisor to the Ministry of Education, Culture, Sport, Science and Technology. He is also a member of the Consulting Committee for ABS Guidelines in Japan and special member of the Nagoya Protocol Committee in Science Council of Japan. With a small background, I request Dr. Suzuki to make the presentation in 15 minutes.
Okay, thank you. However, sorry, I cannot share my presentation. Would you mind turn on my presentation button? Okay.
Okay, we will share it for you. Okay.
Okay. Okay. Can you hear me and can you see my slide?
Yes, yes, we can. And
Okay.
you're audible too.
Okay, yeah, thank you. I will start. Okay, first of all, I would like to thank the Dr. Sharma and Dr. Lili and the ANRRC committee member for the opportunity to make this presentation. And I also listened to the morning presentation by Dr. Philip Desmes. And I totally agree that his opinion about the biobank as a pivotal role and the core of innovation. And also, I will skip the explanation of the CBD and Dango protocol itself. My main topics today is about the digital sequence information, DSI, which is currently under intense discussion at the Convention on Biological Diversity. Okay, COP13 in Mexico 2016, genetic information, genetic information represented by DNA sequence was named DSI, digital sequence information. DSI is a temporal name, and also it has not been determined from where to where. Area of the DSI and definition of DSI. However, some countries require that it should be included in genetic resources and should be subject to the benefit sharing for the use of the genetic resources. Because some country is worried about concern about the creation of the genetic resources from nucleotide sequence data without prior permission. And at COP14 in Egypt, Sharm el-Sheikh 2018, the COP decided by decision 14 establish a scientific and policy process to resolve the differences. And the discussion is going on. And now topics is focus about the handling of nucleotide sequence data in public database, and also CBD secretary propose a policy option and criteria. In next year, 2022, April in Kunming, COP15 will be open, and some kind of decision will be made at COP15. Okay, and from CBD seminar, I would like to explain about why do we need to discuss DSI. DSI data, it's not my opinion. From CBD seminar, DSI data is increasing, and researchers now have access to genetic resources from public database and can obtain genetic resource without physical access. And also historically, Nagoya Protocol and Aichi Target were adopted in 2010, 11 years ago. And also on the post-2020 framework, party will not, Africa and another country will not agree to the post-2020 framework until the DSI discussion is settled. This is a policy option which CBD secretary proposed. Option 0 is status quo. It means current status. And also option 1, DSI equal genetic resources. DSI fully integrated to the genetic resources. And option 5 is DSI is not considered equal to genetic resources. And also, option 4 is technical and scientific cooperation is important. And also, yeah, the CBD secretary proposed 5 options, and in addition, African group proposed another option. Another option of the share of the monetary 1% benefit sharing of the monetary profit benefit. And also Mexican government is insist on the track and traceability. This is the criteria. Criteria for the multi-criteria analysis for the consideration of the policy option for DSI. For the saving time, I want to explain each criteria, but I think cost effectiveness and also feasibility is important. And in this month, COP15 Part 1 was held in Kunming. A ministerial meeting was held. Leader of China, Russia, France, UK. Emphasis of the importance of the biodiversity conservation, and also Kunming Declaration was adopted. In Kunming Declaration, emphasis of the global commitment to the ambitious post-2020 biodiversity framework. And however, in statement of Kunming Declaration, data sequence information, the word of the term of the data sequence information is in it. This is a schedule and time schedule from COP14 to COP15. Now, through the report and many webinar and many meetings. Now we are informal advisory group. In informal advisory group, we are discussing. And in next January in Geneva, post-2020 working group will be held. So that we have to discuss about our opinion, and also we have to, we have to discuss what we have to do. Okay, and let's move to our activity. This is our institute, the National Institute of Genetics, and we have 3 basic science infrastructure, such as the DDBJ and Bioresource Center and Advanced Genomic Service. And under the NBRP, National Bioresource Project, now we are working as an ABS support team for academia. Okay. And we are the ABS support team. We have a whole mission to support the implementation of the Nagoya Protocol and ABS for the university and academia. Mission A is to support researcher and the research community in acquiring the genetic resources by awareness raising and providing information and direct consultation. And mission B is to support Japanese university in building ABS systems. As Mission C, we support NBRP, National Bioresource Project Institution, to implement ABS. And Mission D is to participate the meeting of the CBD DSI. The response is to the Mission D. As Mission C, we support the NBRP, National Bioresource Project Institution, for ABS implementation. And we have conducted the survey of all NBRP institutions to ensure that no sample with ABS problem have been distributed. We have checked the genetic resources since 1993. And we continue to support the NBRP in implementation of ABS, including the individual consultation. This is our recommendation to NBRP institution. Collection, deposit, distribution, and in general, each stage and in general, it requires a precaution described here. And how to deal with the DSI will be the future discussion. At least country of origin. Description of the country origin will be more necessary. We need to set up a system to deal with this. And we also support the make of the recommendation from the Science Council of Japan. From Science Council of Japan, submit the recommendation to the CBD Secretary. Recommendation 1 is the use of the DSI should not be included in the framework of the CBD and Nagoya Protocol. And recommendation 2, restriction should not be placed on the publication and use of DSI. Recommendation 3 is a priority should be given to the facilitated access to genetic resources. And also last recommendation is the scientists all over the world should join the discussion. Yeah, in conclusion, okay. At COP14, DSI, some country require of the DSI should be included in the genetic resources, and some country request is a subject. It is a subject to the benefit sharing for the use of genetic resources. And now we are discussing toward the OEWG3 in Geneva in next January and COP15 in Kunming. And also providing country want to the benefit monetary sharing and using direct genetic resources, and require the traceability and benefit sharing system for public database. However, I worry about the balancing. Okay, today's morning seminar by Dr. Philip Desmes says the balancing of the monetary benefit, non-monetary benefit sharing and openness, and also he said I agree and especially ex situ collection such as the culture collection biobank are also being discussed. so that we have to discuss about what is the importance. My opinion is that science is important, and also balancing of ABS and DSI is also important. And I want to emphasize the need for the clarification of definition and scope of DSI and feasibility of ABS system for DSI. And we need evidence-based discussion of cost and benefit. Yeah. And I don't want the CBD and Nangor protocol to make it difficult to use the bioresource. Yeah, I would appreciate it if you could help me. Yeah, your opinion is welcome. Thank you.
Okay, thank you very much, Dr. Suzuki, for making the presentation time. There could be some questions
yes.
In the O&A. I have not been able to find out. Lily, can you help me in reading out the questions? There could be some clarifications, questions from the participants.
Right now, there is no question yet in the Q&A, but maybe from the audience.
Okay, yes. Any question from the audience?
Philip, you have any question? Maybe
Well,
the quick one, because it's been late already. Just maybe one
surely.
question.
Well, I have no question. Yes, of course, I have a lot of questions, but I fully agree with the presentation of Professor Suzuki, and I would add on that maybe we are missing those that should get the benefit sharing. It's first of all the scientists. I think that we should focus on providing the scientists of source country with the tool to exploit the so-called DSI, what we call the nucleotide sequence database. What the source country needs is instrument and training to get their scientists exploiting the nucleotide sequence database. Then there is a clear benefit sharing.
Yeah, thank you. Yeah, thank you. And capacity building. Yeah. Capacity building is important. Yeah, I agree.
And more important is that different countries have to come on some platform. Isn't it? There's a lot of discussion. There are a lot of difference in opinion among different countries on, on DSI, and also the implementation, as I said, of the Nagoya Protocol in different Asian countries is at the different stages, isn't it? So now these issues are very important, really, that we agree, we harmonize, these systems so that countries can be benefited from the exchange of these bioresources from other countries. So if there's no other question, then I must thank Sunil Archik and Professor Suzuki for making very good presentations, and more importantly that they completed the presentations in time. Thank you very much, and I also thank all the participants for listening to these presentation very patiently. Thank you very much.
Yeah, thank you very much.
Thank you, Professor Sharma, and all the speakers of this session. So as the next session will be less than 30 minutes, so I will skip the break, and before we proceed to poster award presentation, we will have a quick group photo session. I would like to ask everyone to have your camera on if you can, please. So it will be quick. Okay, so on my count, 1, 2, 3. Is it like that? Okay. One more time. 1, 2, 3. Okay, thank you very much. And now, ladies and gentlemen, it's time to announce the winner of Poster Award 2021. And I have to say that it was a tough decision for our judge today, as all posters not only have very insightful scientific content, but also are nicely prepared with visual appeal. So the judging criteria is based on the scientific merit, materials, and the flash presentation. And the best poster award of ANRRC 2021 goes to Mikiya Endo, okay, from Riken BRC, Japan. Okay, and for the first runner-up, we have two first runner-up. The first one is Supatra Kitikun from Biotech, Thailand. And the second first runner-up is Dr. Panyapon Pumkao from Biotech, Thailand. Okay, and the second runner-up is Dung Thi Lei, Institute of Biotechnology, Vietnam. And the last one, the popular vote, go to Arisaldo E. Castro from University of Philippines, Diliman. Okay. So we congratulate them all, and the e-certificate of their awards will be sent to them right after the meeting. Thank you very much. Now, to officially end the ANRRC 2021 conference today, please welcome Dr. Toshiko Shiroishi, the current president of ANRRC and the director of Riken Bioresource Research Center, Japan, to deliver closing remarks. Please, Dr. Toshiko.
Hello, everyone. I am Toshihiko Shiroishi, Director of Riken Bioresource Research Center, Japan. I succeeded directorship of the Riken BRC from Dr. Yuichi Obata, a former director, in 2 years ago, 2019. And at the executive board meeting, in the last meeting, which was held in the Los Banos, Philippines, I was appointed as a president of ANRRC, succeeding a former president of Dr. Liu of Institute of Microbiology, Chinese Academy of Science. My term of presidency just started at this meeting this year, 2021. Okay, so first of all, on behalf of the ANRRC board members, we thank Lily and all her colleagues of National Science and Technology Development Agency, NSTDA, Thailand, for hosting this international meeting this year in this difficult time. This is the first online meeting in the history of ANRRC. It was very well prepared, and the online meeting worked very well, I think. The oral presentation by invited speakers and the poster session were both very successful. I appreciate all invited speakers for wonderful talks. And also, I thank a poster presenter for their beautiful posters. We also thanks everyone who participated in this meeting online. We congratulate all the winners of Poster Session Award. We hope that this award will encourage all winners to further move forward in the research and the research resource-related enterprise. I am convinced that this research resource or bioresource are most important infrastructure for the life science and innovations. And without these resources, the science and industry would not make any progress. I think that the mission of bioresource centers, especially in Asia and Oceania, is very important. This is because these regions are hotspot of biodiversity in the world. And at the same time, these regions are economically rapidly growing. This means that we have a great potential and a huge advantage in promotion of science and industry, including biocircular green economy, BCG economies, which is the main theme in these meetings, based on bioresources. Influence of our activity on the worldwide science and economy is getting larger and wider. On the other hand, one concern is the decline in biodiversity due to economic growth. Given these situations, I think we have a very heavy responsibility for facilitation of use, sharing, and preservation of bioresources across national borders. Therefore, we need to communicate and share the management system for bioresource enterprise between different bioresource centers. And we need more cooperations and tight alliance among all member countries to achieve our goal of ANRRC. Finally, I have one more important announcement. Today, ANRRC board meeting decided that in the next year, the ANRRC 2022 will be hosted by RIKEN BRC, our institute, at Tsukuba, Japan, in the same season, probably October or November. Unfortunately, it is difficult for us to predict what the COVID-19 pandemic will look like around the season next year, and how it would be easy for participants to enter Japan. So therefore, it is not clear at this moment whether it can be held in person. I know that many people wish to have the meeting in person, but actually, we cannot say at this moment, it will be held in person or it will be held as an online meeting or in a hybrid format. I think we can announce more detail in the next spring, maybe around March or April. Anyway, I look forward to seeing all of you next year again in the ANRRC 2022 meeting. Okay, I stop here. Thank you very much for attention.
Thank you very much, Dr. Toshihiko. Okay. Okay, so before we leave, I would like to inform you again that the posters will be collectively presented as e-proceeding and can be accessed on the ANRRC 2021 webpage, as well as all the slide presentation today. So finally, on behalf of NSTDA, I would like to express my warm appreciation to all executive board members, distinguished guests, speakers, chairs, and all participants for your efforts and kind support in making this event a success. So we are hoping to see you all next year in ANRRC 2022, which will be hosted by RIKEN Japan. So thank you very much and goodbye.
Thank you.
Yes, thank you. See you next year.
See you.